video is Phillips, and today we'll be looking at plant physiology. This will not include plant hormones, which will be on a separate video. So if you're looking at tropisms, please find that video. So just to introduce the idea that organs are found in plants as well. So we know organs are a group of tissues working together to perform a same function, and the organs will contain more than one type of tissue. And plant organs include the leaf, the stem, the roots, and the flowers. So you should be able to organise some tissues and organs in plants and what their role is. So the flower you should know is a reproductive part. The stem is going to be supporting the leaves of the flowers. The roots absorb the water and minerals, and the leaf carries out photosynthesis. From this one, you could say the organ is the leaf, and tissues could be the spongy mesophyll. So in terms of plant organs then, there are different parts of the group working together, and tissues of the plant include the epidermal tissues, the mesophyll tissues, the spongy mesophyll, the xylem, the phloem, and the meristem tissues as well. You need to know the different positions of the leaf, so if you want to pause this and have a go, feel free. But here's the answers. So the leaf starts with a waxy cuticle, which is a waxy layer preventing water loss. We then have the transparent upper epidermis, which allows the light to transfer through into the palisade mesophills, which have a large surface area and are densely packed with chloroplasts, allowing photosynthesis. We then have the spongy mesophyll surrounded by air spaces that allows the gases to diffuse in and out from the stomata at the bottom of the leaf, allowing photosynthesis and respiration. The lower epidermis, which contains the guard cells, which open and close to allow the stomata to open or close. So in just identifying the functions of the different areas, so if we go through, we have the upper epidermis as a top layer of cells made of transparent cells to allow the light to get through. The cuticle is a waxy layer on the leaf surface, preventing water loss, will be thicker in xerophytes. Chloroplasts are the sacs containing, or membrane sacs containing magnesium, which makes chlorophyll and absorbs wavelengths of light to do photosynthesis. The palisade layer is the layer containing most chloroplasts, and they're doing most photosynthesis. The spongy mesophyll are layers with the air spaces in them. The vascular bundle is a term we haven't come across yet, which is where the xylem and phloem vessels are found to transport water and minerals and sugars around the plant. The stomata are the holes, mainly in the underside of the leaf, to prevent too much water loss, which allows gases to diffuse in and out of the leaf and are involved in transpiration. The guard cells open and close the stomata to control water loss. The lower epidermis is the bottom layer of the leaf, and the air spaces are areas allowing diffusion of gases through the leaf to the palisade mesophyll cells for photosynthesis. So, transpiration, and we need to know what this term means. It is the loss of water from evaporated from the aerial parts of the plant. So the water is evaporated in the leaf and lost through the stomata. And basically water builds up in the mesophyll cells and evaporates to form water vapor. It builds up in the air spaces of the spongy leaves and then diffuses out due to the fact that it is vapor, not water, so we say it's diffusion rather than osmosis. If the stomata are open, this is very easy to happen. So what happens is the osmosis occurs from the xylem to the mesophyll cells. The sun shines on the mesophyll cells, causing them to evaporate, and the water vapor appears in the air spaces and the spongy air spaces, and then diffusion occurs down a water vapor potential gradient from the inside of the leaf out via the stomata. There are two main factors that affect the rate of transpiration: temperature, because the higher the rate of temperature, the higher the temperature, the higher the rate of evaporation, and humidity, because if you've got a low humidity in the air, it means there'll be a high concentration gradient between the inside of the leaf where the water is evaporated and the outside. If the air is humid, that means there'll be a lower concentration gradient between the water vapor inside the leaf and the humid atmosphere outside the leaf, so there'll be less water loss as a result. So what happens is, as the water evaporates out of the leaves, this draws more water up the xylem via transpiration. Water is lost though through the stomata. Okay, so the stomata obviously are opening to allow gas exchange, but they will close when they are hot to prevent excess water loss, and they will close in the night time because plants cannot do photosynthesis at this point, so the stomata do not need to be open. So these are two things that can also reduce the rate of transpiration. So the factors that impact the rest of transpiration is temperature. Obviously, higher temperatures will lead to higher energy, so the particles can diffuse faster. But if the plant gets too hot, it will close the stomata. Air resist movement due to wind. The more air movement there is, the more water will be lost as the water will be blown away, maintaining the concentration gradient. The lower the humidity, the greater the concentration gradient between the air, water, vapor in the leaf and the air outside. That increased in diffusion will mean more water is lost, and the more the light intensity, the more photosynthesis, so the more the stomata will be open for diffusion. So let's look a bit more at transpiration. If you think about the size of leaves, trees, and it's incredible how far the water is pulled up through the plant. We couldn't achieve this through pumping mechanisms. So plants have got incredible techniques to push water up to the leaves via transpiration. So transpiration occurs due to a combined effect of these four things: cohesion tension between the water molecules sticking together due to hydrogen bonds, adhesion, where the water molecules stick to the side of the xylem, preventing them moving back down, root pressure, i.e. there's high pressure at the roots and low pressure at the leaves, so it forces it upwards, and the fact the xylem vessels are so thin, maintaining the number 2 force, adhesion. So if we look at this diagram, it shows you what exactly happens. So as we said earlier on, the water here in the mesophyll cells evaporates due to the heat. It builds up here as water vapor and then diffuses from a high concentration inside the leaf to a low concentration outside the leaf. By the water evaporating here, that lowers the water potential in these cells. So then water moves on a high to a low concentration here, and again from here to here, setting up a water potential gradient. This means the water pressure here is low because water is leaving, and water entering from the root is high, so you get a high to low pressure as a result. So water must go through the leaves and the root cells before entering the xylem. Instead, you get a high pressure at the bottom, low pressure at the top, causing a change. Transpiration will not occur with high humidity because that means you don't have a high concentration here and a low concentration here. Instead, you have a high concentration outside. At night time, these stomata are closed, and in the winter for the deciduous trees, the leaves are lost, so this would be blocked off here as a result. So that is key bit there. The pressure, as we said before, the xylem pressure, the root pressure is essential. It gives high pressure, pushing it up the leaf. The xylem structure of the xylem is adapted by having something called xylem vessel elements, which is a proper cell name, and they carry water insoluble minerals up the cells, up the plant, because they're long walls. They're impregnated with lignin, which is a waterproof layer, and the whole cell dies when it's mature, which means it can be hollow. The lignin gives it strength and stops the walls collapsing when the pressure drops at times during the day. The lignin can be laid down in different patterns, allowing different shapes and branches to come off the xylem. So the xylem is dead in a continuous column when they're mature. They're very thin tubes, so the water column doesn't break easy. Any pits in the lignified walls allow sidewards movement if the xylem gets blocked, and the lignin allows the bending and stretching of the plant. The key thing is the water is not impeded because there are no end cells, no contents of the cells, they're hollow because they're dead, including no nucleus, and the lignin prevents collapse when pressure changes in the cell. So as said before, you mainly have evaporation via the stomata. Water from these cells here evaporates to form vapor, and then you get a concentration gradient from here, from the xylem across the leaves. So how does the water move up the stem? As I said, it's due to root pressure, which I talked about earlier, but also something called transpiration pulls. So the low pressure here and the water potential means more water drawn across, and low pressure. but also the water sticks to the side of the xylem called adhesion, and the water molecules themselves have hydrogen bonds between them, holding them together like a chain called cohesion tension theory. So you can see that here, the cohesion tension theory, these water molecules will be pulled together, so as you pull them up, it's like a string of water molecules being pulled together, so because of this hydrogen bonding here. And the water molecules adhere to the side, stick to the side of the tube because of adhesion theory. We know that this happens because in the daytime, when there's more pull on the xylem, there's a negative pressure, and tree trunks will have a narrower diameter as a result. When the nighttime comes, this pull is stopped, and tree trunks will have a wider diameter because the pressure is no longer negative. If the xylem vessel breaks, the water can no longer be drawn up, and therefore the pressure is also lost. Now we can measure the rate of xylem, measure the rate of transpiration using something called a potometer, which is very simply shown here. And it makes a big assumption that the water uptake in a plant is the same to the water lost via transpiration. Now we know this isn't true, it's about 99%, however, so it's an assumption made with all potometers. Now you would be expected to say how you would set this up, and you would have to do it underwater, so you would put this part of the plant in underwater to make sure there's no air bubbles in the apparatus. You would ensure the tube is tight by using petroleum jelly or Vaseline that seals. You would need to cut the stem at an angle to ensure water uptake and increase the surface area. You control all of the conditions other than the thing you're investigating. So if you're investigating temperature, you would control light intensity, carbon dioxide, etc. And you would then measure the movement of this meniscus up the potometer to work out the volume of water uptaken under different conditions. So you might be given something like this in an exam where you would measure the volume of water, you'd measure the length, and then you can work out from that length what the actual volume of water is by doing pi r squared times the length that the water moves as a result. Translocation is the movement of sugars or products of photosynthesis, or sucrose to be precise, up and down the plant from sources to sinks, and it occurs via the tissue of the phloem. The phloem is a tissue because it's made up of two cell types. They are sieve tube elements, which are the tubes that the sugars transport through. They have no nucleus and very, very little cytoplasm, so sometimes are not called true cells. They line up end to end, and they have little holes in the cell walls at the end called sieve plates, which allow the sugars to move through, or sucrose to move through. They are normally six-sided in hexagonal shapes. They are attached to something called a companion cell. Now, the companion cell has a dense cytoplasm with lots of mitochondria, which do the active transporter and the active processes, for the sieve tube elements. Effectively, they are like a life support machine, keeping the sieve tube elements alive, even though they don't have all the components in the cell cytoplasm. And they are linked by a continuation of the cytoplasm called plasmodesmata. So translocation is the movement of organic substances that are made in photosynthesis generally, and that is normally sugars as sucrose. And basically what happens is the sucrose moves from the source, the area that it's been made, to the sink, the area that needs it to be made. And this is done by a couple of stages. First of all, the sucrose is actively loaded into the phloem. Now how it does this is it builds up in the leaf cell. Sucrose then diffuses into the companion cells before hydrogen is actively transported out of the companion cells and into the cell wall here. Then the hydrogen diffuses into the sieve tube, and it then takes the sucrose with it by a co-transporter protein. We then have something called the mass flow hypothesis, which is that because we've now actively pumped sucrose into the xylem, it lowers the water potential into the phloem, it lowers the water potential, so water moves in from the neighbouring xylem, increasing the pressure in this area, meaning it has a high hydrostatic pressure, and that moves it down to where the sucrose is being removed, where it has a low hydrostatic pressure. So you need to understand that sucrose is actively loaded at the source. This lowers the water potential in the phloem sieve tubes. The water then moves from the local xylem in via osmosis, raising the hydrostatic pressure. At the sink, the sucrose is used in the local cells for respiration, so it is moved via active transport. The water potential, therefore, in the local cells also drops, and the water follows out via osmosis, creating a low hydrostatic pressure, meaning the hydrostatic pressure in the source is high and the hydrostatic pressure in the sink is low. So is this active or passive? Well, mass flow is passive based on the hydrostatic pressure differences, but it obviously relies on active loading of the sucrose into the phloem sieve tubes and the active transport of sucrose out of the phloem sieve tubes into the sink cells. And the pressure differences are the key thing that causes this difference, and it can happen in either direction. It's always a higher pressure at the source and a lower pressure at the sink, regardless whether it's going up or down to get to them. Now, some evidence for mass flow hypothesis can be seen. We can follow where sucrose goes by radioactively labeling carbon dioxide, which then becomes the glucose, which then becomes the sucrose. You can puncture the phloem using aphid stylets, and then it will ooze out under pressure, showing you there's a high pressure as a result. And we can also record the fact that during the day, more sucrose moves around the plant, showing the energy is needed, which is gained through photosynthesis and then respiration. If we use something called ringing of the trees, we can see that the phloem bulges above the cut, showing you that the sucrose is moving in the phloem on the outer part of the stem. So now we're going to look at sexual reproduction in plants. So plant section of the flower the flower. And plants can reproduce sexually because they have gametes just like animals do. Instead of egg and sperm, we have egg and pollen. And instead of an embryo, they develop a seed. Uses insects, and they're essential for our sexual reproduction of plants. The insect is attracted to the brightly coloured flowers or the smell of the plant, and they go to get the nectar. But when they do, they rub against the pollen, and therefore they take the pollen to the next plant and give it to the next plant. But we need to know that in a bit more detail. So to understand this, you need to know the parts of the plant. So as we look at the parts of the plant, we have the stamen, which is the male part of the plant, and that includes the anther, which produces the pollen and is held up by the filament. So when the bee lands on that, the pollen rubs off onto the bee, it then flies to another plant where it lands, and the sticky stigma holds onto the pollen. The pollen then grows down this thing here called the style, and it goes into the ovary where the gametes fuse to make the egg, be fertilized to make the zygote. So there are a couple of types of reproduction. There is insect pollination, and you need to know how the plant is adapted to being pollinated by insects. So they'll have brightly coloured plants with sweet smelling petals. They'll often contain nectar, so the insect will eat it. Small quantities of pollen being produced because they know they'll stick to the insect, and it's sticky or spiky to stick to the animal. Their anthers and stigma are really held in place in the flowers so the insects can brush against them, and the stigma has a sticky coating to accept it. Wind-pollinated plants, however, are different. They don't rely on insects, so they'll often have small petals so the wind can blow at them. They won't produce nectar because they're not trying to attract insects. The pollen is produced in huge numbers and is very light, so it can a very low mass, so it can be blown far in the wind. The anthers are loosely attached and dangle out of the flowers to make it easier to release the pollen, whereas the stinger hangs outside the flower to help it catch the pollen as a result. So you need to be able to compare the two different wind pollinated and input pollinated and explain it. We can also talk about cross-pollination and different methods of it and why it's important. So cross-pollination is the transfer of pollination pollen even from the anther of one plant to the stigma of the other. It leads to the fusion of gametes, leading to genetic variation and evolution through natural selection. This allows hybrids to be created and creates hybrid vigor. This is when two unrelated plants breed and they are healthier and grow stronger because of possibly more heterozygous alleles, so different alleles for each gene as a result. And you can see here corn plants have been inbred for species A, species B, and when they're bred together, you get that hybrid vigor, stronger, bigger plants. Inbreeding, however, does happen in plants. Many plants are hermaphrodites. They produce both pollen and eggs, as you've just seen. So self-pollination is possible from transferring the pollen and anther from the stigma in the same plant. This can lead to inbreeding depression, which is more likely that a rare negative double recessive allele will be inherited. Some plants have evolved methods to prevent this. But despite prevention methods, it does occur. And many plants will not actually germinate. They'll be self-incompatible as a result, and this increases the genetic variation. So what will happen is if they pollinate themselves, the pollen tube will stop growing and it won't become a zygote as a result. This means that there's more chance of another plant pollinating that plant, and it means more variation as a result. There are many self-incompatibility mechanisms involved, but basically the idea is they will not allow self-pollination. You also need to be familiar with seed dispersion mechanisms, which allow the seed, which remember is the embryo of the plant, to be spread as far as possible. So wind is a method of taking far. Peas will explode under high temperatures. Gravity will then take these things further down. Animals might eat the seeds, they won't be digested. The animal will then move from one place to another and carry the seeds and release them in the feces, which will then grow further afield. Same for insects and birds, and pollination for insects obviously links to that as well. Water, and if they fall in the water again, they will spread as a result.